Dear students, you can purchase all the lessons in A-level biology and chemistry of the local syllabus in Sri Lanka, English medium and Sinhala medium. Refer the catalogue of this WhatsApp number and the description box of this video for more details.
7. Molecular biology and recombinant DNA technology. Initially, I mentioned at the beginning of this lesson, I mentioned this lesson can be divided into two parts, molecular biology part and the recombinant DNA technology part. Under the molecular biology part, still we are in the molecular biology part, then mutations. Yes. According to your knowledge, what is the term mutation? Mutation means, simply we can say, mutation means a change. It is a change from the original version of something. It is the change of the original version. In these mutations, we say the change is a permanent change. Permanent change. So what is the term phenotype? I think you did the lesson genetics, unit 6. This is unit 7. After doing unit 6, you know about this term phenotype. Phenotype means what? Phenotype means, we learned, phenotype is the output of the genotype. Phenotype is the output of phenotype. Phenotype is actually the character. Performance of the, it should not the performance. After performing the character, we can see the phenotype. For example, eye color. Then eye color means blue eyes, then brown eyes, dark eyes, then so many eye colors. Like that, the phenotype means the outcome, output of the genotype. Genotype means what? Genotype, it can be explained as the genetic code, and which is stored in gene. It is the genetic code actually. So usually we say phenotype controls the genotype of an organism. Phenotype controls the genotype of an organism. But other than the phenotype, there is another factor which controls the phenotype of an organism. Imagine the skin color. Skin color of African countries and European countries. What about the difference? Dark skin. In African countries, we can see comparatively dark skin in humans, but in European countries, it is not too much dark. The skin is not too much dark. Why? These skin colors are different in such a way due to the weather. Especially in African countries, they have to protect their body from high intensity of light. That is why they have a dark color skin, rich in melanin. When melanin is rich in the skin, it becomes dark skin. Therefore, the phenotype is the final outcome of the genetics or phenotype and the environment. Okay? There are two factors are involving to determine the phenotype of an organism. First one is what? Interaction between genetics, and second one is effects of the environment. The phenotype of an organism basically depends on the genetic information stored in DNA, and the final outcome is the result of the interaction between genetics of the organism and the effects of the environment. Alternation in DNA may bring about certain changes in the characters of individuals in a species, which result in phenotypic variation among organisms. These changes occur permanently, permanent changes of the phenotypic genotype, permanent changes of the genotype, therefore phenotype also differ, also it's changed. So permanent changes, these permanent changes are called mutations. Permanent changes are called mutations. Mutation means an alternation of the nucleotide sequence of the genome of an organism. That is why I mentioned it as changes, permanent changes of the genotype or the genetic code or the genome are called mutations. Okay. Mutations are the sources of variation seen among individuals of a species. The effect of a mutation may be neutral. And some mutations may neutral. Neutral means what? No considerable outcome, no considerable difference of the phenotype. And some mutations may beneficial. Actually, beneficial mutations, they can actually the beneficial mutations are so much lower than deleterious or harmful mutations. Many mutations are harmful, deleterious. Deleterious mutations may be lethal, or at least they are less favorable than the original phenotype. A mutation may also cause complete, remember that the mutation may also cause complete loss of a function if the gene controls the vision of the eye. If that particular gene is mutated, it may cause a complete loss of the vision, which means blindness. A mutation may not always. A mutation may also cause complete loss of a function. In rare occasions, the function of function of the polypeptide, which means a protein, may be enhanced due to mutations. In rare cases, they are beneficial. And these are beneficial mutations. Previously, we learned genes control characters. Genes control characters. What are the characters actually? Examples: eye color, skin color, the vision. So many characters we can see in our body. Each character is controlled by a gene or set of genes. Characters are controlled by genes by producing proteins. How proteins can be made? By making polypeptides. After making polypeptides, we learned it, the synthesis of polypeptide. After making the polypeptide according to the genetic code of the gene, they may modify it into the specific protein, into the quaternary and tertiary structure. Anyway, these mutations are occurring in the genotype. Some are beneficial. Rarely they are beneficial. Most of them are harmful, which means deleterious, harmful, lethal, or less favorable. The beneficial mutations are very rare. Some are neutral, no considerable effect. Entirely new functions may also be resulted by mutations. As the gene makes very dark color skin, after mutated it, the gene may produce very light skin. It may reduce the number of pigments, number of pigment molecules, like that. Entire new functions may also resulted by mutations. New functions means there may some patches in the skin, like that. For example, an enzyme specific to a substrate may here drastic change. Enzyme specific to a substrate may change due to mutation in such a way that is actually enzyme specific to a substrate may change due to mutation in such a way that it changes its specificity to act on another substrate. Like if DNA helicase, right? Helicase is made by gene. Right? After it is mutated, it may produce, but DNA ligase like that. Right? The product due to the mutation is capable of catalyzing a new biochemical reaction. Then enzymes, the relevant product is changed. Therefore, as the product is an enzyme, then it may catalyze a new biochemical reaction. Mutations are the sources of variation seen among individuals of a species. There are two major types of mutations based on the scale of alternations in the genetic material. There are two major types of mutations based on the scale of alternations in the genetic material. First one, small scale changes. Second one, large scale changes. Small scale changes, large scale changes. Small scale changes are gene mutations. Large scale changes are chromosome mutations. Sorry. Small scale changes are gene mutations. Large scale changes are chromosome mutations. Small scale changes occurring in the sequence of nucleotides in a gene. Right? If you can remember, we discussed those things. As this is the chromosome, DNA is included inside this. As this is the chromosome, DNA is included in this. DNA is included. DNA. When we enlarge a portion of this DNA, we can see, when we enlarge, we can see the double helix nature. It has two strands, two antiparallel strands. The first one 3 to 5 and other one is 5 to 3. In this DNA molecule, a small specific sequence of nucleotide, this region we can call it as a gene. Usually this gene controls a character. Genes control characters. When the entire chromosome means number of chromosomes may change. Number of chromosomes may change, as well as the structure of this arm may broken. This arm may break. So such mutations can be occurred. Mutations are occurred actually. Such mutations are large scale changes, which means chromosome mutations. How chromosome mutations are identified? The number of chromosomes may differ. Number of in each species, they have a specific number of a set of chromosomes. In humans, we have 46 chromosomes in our diploid cells. And when the 46 chromosome become 47 or 45, the chromosome number is changed. Such mutations are called chromosome mutations. And under this chromosome mutation, this one also can be considered. This arm is broken, or a new part of another chromosome is attached to this one. This again, it is mutated. This chromosome is mutated. So such changes we called as large scale changes in the number of chromosome or the structure of chromosome. Such changes we called as chromosome mutations. Small scale changes. Small scale changes means inside this chromosome, we can say DNA, very small region comparatively. And when we consider the length of the whole DNA, the length of the gene means very small, very small. When the mutation is occurred inside a gene, such changes are small scale changes comparatively. When we consider it as with the large scale changes, chromosome mutations. So when gene mutations are occurred, they are called small scale changes. Small scale changes occurring in the sequence of nucleotides in a gene known as gene mutations. First, gene mutations are small scale changes. The permanent alternations of DNA sequence. Again and again, remember that mutations can be divided into two: chromosome mutations and what? Gene mutations. Chromosome mutations are large scale mutations. Gene mutations are small scale mutations. Chromosome mutations are two types depending on the number of chromosome or structure of chromosome. And gene mutations, here we are in the gene mutations. And if you can't remain it, mutations can be divided into two. First one, chromosome mutations. Second one, gene mutations. Chromosome mutations can be seen as structure of the chromosome mutations and number of chromosome mutations. Gene mutations are small scale changes. Chromosome mutations are large scale changes. Here we are in the gene mutations. Gene mutations. Gene mutations, the permanent alternations of DNA sequence of a gene, are called gene mutations. They may occur due to rare errors. Rare errors occur during DNA replication. These are called spontaneous mutations. Remember that gene mutations are called as spontaneous mutations. Gene mutations are called spontaneous mutations. In addition, certain external factors can also cause mutations at a higher rate. Since these factors generate mutations, they are called mutagens. Mutagens means mutation occurring agents. Mutation occurring agents like chemical factors, physical factors are mutagens, like X-ray, UV are physical agents for doing the mutations, which are the physical mutagens. Chemical mutagens are in these days we usually talk about cancer causing agents, chemicals in, especially in foods, the instant noodle inside in the packet of instant noodles, like that, such chemicals are called as mutagens. Definitely they may cause mutations. Mutations means finally cancers. Mutagens. The mutagens, here we learned about mutagens. Mutagens means mutation occurring, mutation causing agents. The mutagenic agents can introduce mutations to replicate in DNA in a cell. Mutations are also causing of carcinogenesis. Carcinogenesis means the process of producing. Carcino means cancers. Therefore, mutations are also doing cancers. Are also making cancers. The production of cancers, this process we can identify as carcinogenesis. Carcinogenesis means cancer, cancer occurring like this. Therefore, mutagens are carcinogens. Carcinogens means what? Cancer causing agents. Mutagens means mutation occurring agents. Not all mutations cancers all mutations are not cancers, therefore not all mutations are not carcinogens. Anyway, all carcinogens are mutagens. Carcinogens are mutagens. These chemicals and radiation needed to be handled with uh, very carefully. We have to deal with the chemicals and radiations, especially chemicals in our food. Like these are small scale mutations. We discussed gene mutations are small scale mutations involving only one nucleotide pair or more than one nucleotide pair. Small scale means gene mutation. To consider as a gene mutation, only one pair of nucleotide or um, more than one, but very less number of nucleotide pair. In the DNA, in the gene region, adenine to consider as a gene. Imagine this. This is the gene region of the DNA. This is the gene region. To consider as a functional gene, this nitrogenous base should be the adenine, guanine, cytosine, thymine. This should be, and this nucleotide pair, this nitrogenous base sequence should be occurred to make the functional gene. According to the base pair rule, in the other antiparallel fragment, this is adenine, this is thymine, this is guanine, then cytosine, thymine. The functional gene is like that. This nucleotide pair sequence should be there. Then imagine this guanine and cytosine. Instead of this guanine, adenine has come, and then this is thymine. Now this is a mutation. This is not the original gene sequence, the nucleotide sequence of this gene, functional gene. Therefore, it is a mutation. Only one pair is altered. These are called point mutations. Always point mutations. For to consider as point mutations, only one nucleotide pair should be changed. Only one nucleotide pair should be changed. There are 3 types of gene mutations, including point mutations. A single nucleotide pair substitution means what? Only one nucleotide pair, a single nucleotide pair substitution. Change one nucleotide pair with another. Change one nucleotide pair with another. Nucleotide pair insertion means addition of one or more nucleotide pairs. As this is the end of this gene, additional adenine thymine pair is inserted this gene. Then it is a nucleotide pair insertion, addition of one or more nucleotide pairs. Nucleotide pair deletions. As this is the end of this gene, this pair is deleted. Now it is not the original functional gene. Removal of one or more nucleotide pairs. When it is one nucleotide pair, they are called point mutations. The substitution of a pair nucleotide is point mutation. You know about the insertion and deletion. Substitution means here the guanine was there, then the cytosine comes here and guanine go there. Now it is the substitution, single nucleotide pair substitution. It is also a point mutation. Nucleotide pair insertions, nucleotide deletions may cause point mutations when a single nucleotide pair is involved, inserted or deleted. Insertions or deletions can be point mutations or may involve more than one nucleotide pair. First, substitution. Substitution means what? Initially, adenine, thymine, guanine was there. This nucleotide sequence was there. Then thymine, adenine, adenine, cytosine was there. Then, what happened? Adenine comes here, thymine go there, thymine go there. And then a substitution. Substitution is happened.